Okay, looking at the gram stain again, a little bit more in depth. Uh, it's a stain that's going to distinguish the gram-positive from the gram-negative cell walls. And as I've said before, gram-positive is going to stain violet or purple because it retains the crystal violet that you put on it. The gram-negative is going to stain red from the safranin, and so it's going to appear pink. And this is used for diagnosing infection and deciding what drugs to use to treat the infections that are caused by bacteria. Looking at the gram stain, there are 4 steps in performing gram stains, and they have to be done in the correct order. First, you put on crystal violet, and you can see there the gram positive on the left. It's going to stain purple, so is the gram negative, because crystal violet is very purple. What's happening is because the cell walls are staining those colors, because the cell wall has the peptidoglycan in it. Now the second step is to use iodine, and again, both the positive and the negative are still purple, and you don't really have a lot of effect with the negative over here, but you do over here going to have more because the dye crystals are trapped here in this positive. The third step is the decolorizer, which is alcohol and acetone. This is where you start seeing differences. The gram positive stays purple, but the negative is going to lose its color. It decolors. And that's because the outer cell wall weakens and the cell loses the dye. But in the gram positive, that does not happen. The last thing is the safranin, and that's a red dye. And it doesn't change the gram positive, but the gram negative is going to stain red, which is going to appear more pink because that red, it was decolorized, now we've stained it with the red. So the order of those steps, you've got the crystal violet, the iodine, the decolorizer, and then the safranin. You put them all in that correct order every time when you're doing a gram stain. Now, some bacterial groups don't have a typical cell wall structure. Mycobacterium and Nocardia don't. These are gram positive, and they have, the lipid there is mycolic acid. So they have a lot of resistance to certain dyes and chemicals. And this is when you would use an acid-fast stain instead, that we'll talk about more later. Sometimes, like a mycoplasm, they have no cell wall at all. And it can be stabilized by sterols, but they are pleomorphic. They can alter their shape or size in response to environmental conditions. Now, a cell membrane, if we describe what a cell membrane is, it's the same thing in bacteria that it is in all your eukaryotes. It's a phospholipid bilayer with proteins embedded in it, and it moves like a fluid, that fluid mosaic model where those phospholipids can move back and forth. It functions because it's the site for a lot of energy reactions, for processing nutrients, and for making a lot of molecules. It regulates the passage of nutrients and waste out of the cell so it can secrete things, and it's selectively permeable. It picks and chooses what can get in and out and when it can get in and out. And this is just a reminder of what it looks like. You see the phospholipid heads and tails, and it's a double layer that you have here. You've got all these proteins, this purple and this green and this lighter colored purple and that blue over there. Those are all proteins that are embedded in the membrane that helps things move back and forth across. And then you've got some cholesterol is in here that will help stabilize the membrane and keep the fatty acid tails from sticking together. And then you've got some identifying molecules here that will identify it so that other cells know what it is there. But inside that bacterial cell, of course, you've got cytoplasm. And cytoplasm is a gelatinous solution. It's got sugars, amino acids, and salts in it. And this is the site for biochemical and synthetic activities. It's 78% or 70 to 80% water. So it's a solvent for materials that the cell needs to function. The nucleoid is the area that contains the chromosome. And in bacterial cells, it's a single circular double-stranded DNA molecule. It contains all the genetic information of the cell. Plasmids are some non-essential DNA. They're extra DNA. It's a little circular double-stranded DNA that's just out there by itself. And they use these in genetic engineering because they can be manipulated and transferred from cell to cell. They can cause drug resistance, and sometimes they even produce toxins.